Well, our next talk this afternoon is by Bob Murphy from CMU, and from the sound of it, he's going to tell us a little bit about active learning in a different context, where the kind of oracle, if you will, where the source of our information isn't a human expert or a crowd of humans, but experiments. And I think this is a really exciting area for active learning, and I'm really glad we have Bob here to tell us about it. So take a look.
Okay, so thanks very much for the opportunity to talk to you. So, of course, nowadays all we hear about is big data, all the opportunities of machine learning and everything. Well, I'm here to talk to you about a problem that is completely the opposite of that. Even though you're hearing all this about bio-big data, and there's so much data coming and there's so much data available, the problem is in biology that we have nowhere near the data that we need, and it is sort of the quintessential example of where we need active learning. And let me just try to motivate that a little bit more in terms of a little bit of biology and medicine. Diseases are, as we know, many of them are quite complex, can be affected by many variables. We each have different genomes, that affects things. The effects of drugs can be different depending on what your genome is, depending on the patient, depending on the history of the patient, and so on. So if you try to imagine a model that you would build that would say, what dose even, let alone what drug, should I give to this patient based on all of these other variables, I would have to measure a huge matrix of all of those variables in order to build a model to tell me what to do for that. And in addition to that, in the drug development space, the biggest problem in drug development is not finding drugs that do what we need them to, although that's an important problem. It's finding drugs that do what we need them to do that don't do other stuff, all right? And so that means, as we'll see in a minute, exploring a very large space of what effect each drug would have on many different things in the body rather than just on the one thing that you're interested in. So that's also a large space. In all of biological research, it turns out that the first thing there is something that I never imagined was going to be true when I was a kid, deciding I wanted to be a biologist. Right? We thought that you would learn the book that I read that convinced me to start out as a biologist. It was called the genetic code, rules, right? You learn the rules. Well, sorry, turns out there aren't any rules. There isn't even a genetic code, it turns out, at least not a fixed one. And so what that means is we're in a space of biological systems that are complex systems, no rules. All that we can really do is build empirical models of all of this stuff, which includes the fact that we have tens of thousands of proteins and genes, and we have hundreds of cell types, and we have all of this is all interacting. It's huge. So let's try to take a piece of that, and let's take a piece of that where we're going to try to learn for some system a matrix or tensor that describes the responses of the system as a function of different sets of variables. And what I'm going to mean by that, and we're just going to stick with this as a motivating example, but we can change the variables as we wish, is let's think of the case where we have many possible drugs, so that's variable number one, and we have many possible targets, that's variable number two. Okay? Now we can add 3 and 4 and 5 as we want in terms of tissues, cell types, patients, genotypes, anything we want, but we're going to just stick with that for right now to try to go forward. All right? And obviously, we want to do that without doing all possible experiments because the order of the number of drugs is in the millions, the number of targets that drugs can have in cells is at least 10,000, probably more like 100,000, and so we're not going to be able to do all those experiments. All right, active learning. Now, let's contrast where we're going to the way things are done now. What happens now is that we pick a particular target some protein important in cancer, and we try to find a drug that blocks that target. So we do a screen for a lot of different drugs for that target. And there's even some active learning work to try to reduce the amount of experiments you have to do for that. But the problem is, as I said earlier, that's not really what we're trying to do. We didn't learn anything from protein 1 that helps us with protein K, and we need to learn the whole matrix, okay? And so people have thought of this problem from two perspectives. One, from a perspective of learning, doing a matrix factorization type of analysis where you try to take whatever you have and you try to build a model from that, or from a perspective that we just want to take data on those drugs we know already, and we'll just try to use that to predict entirely the behavior of other drugs that we haven't measured without doing any experiments. So those are the two extremes. Neither of those are really satisfactory. And so what we want to do in sort of thinking about what kind of active learning problem is this, is to think about these considerations. So the first is that unlike almost all the active learning examples that you will see, in this space we have no unlabeled data, right? So there is no data that we just need to know the label of. There's nothing, right? All we have is the variables that, all the different drugs I could have and all the different targets I could have. I know what they are, but I don't have any data I don't have anything at all other than the response that I measure of one of those targets to one of those drugs. But I have to do an experiment to get that response. So it's not like the case of unlabeled documents or something, okay? And we are typically starting with very little data. And when we start with little data, if we consider how much data is missing, if only a little bit of data is missing, okay, fine, we do some matrix completion, and we predict whatever is not missing, and we're going to probably be okay. And in the case that I mentioned where we have data, a lot of data, all the data on some drugs and not for other drugs, then we do some kind of factorization and we say, okay, we're going to predict for the new ones. That's been done reasonably well. But it turns out that the real case that is important is that most of the data or all of the data is missing, okay? And we have no data, so we're starting basically from nothing, okay? Or from very little. Now, in that case, we might still have some information to try to make predictions, which would be features of the drugs or the targets, okay? That is, measures of, for example, the chemical similarity between the drugs, or measures of how similar the pathways are, the two different targets are within the cell, whether or not they affect similar behaviors or something like that, right? So maybe we do, maybe we don't, right? One of the problems is even when we do, we don't necessarily know how good they are, right? So that's another whole issue. And then the other thing we need to consider is what is it that we are trying to predict, right? In all of the active learning that's been done, very little of it that has been done in biology, it has been done where we're trying to predict basically a binary value and if sometimes a real value, okay? But it turns out that for many responses, we can't think of it in those terms. That is, there's not a single axis over which the response occurs. So that I can take a drug and it might affect my heart rate in one patient, but affect my clearance rate for other drugs in another patient. Those are two totally different effects. So what I want to look at there is the class of the response, as well as potentially some magnitude. But mostly I'm thinking in terms of the class of response. Does it affect my kidneys, yes or no? Does it affect my heart, yes or no? Does it affect the transport of some protein in the cell? So that's to just frame where we are with the problem. Okay, so the other thing that we're facing and we've had a number of talks about active learning already, that almost all the work on active learning, there are some exceptions, is done to sort of test an approach using retrospective studies where you already have the data available, and you even can cheat a little bit and say, well, based on what I know about the data that I have available, it meets the assumptions that I'm making in order to try to build my model. Oh, yes?
Can I ask you a question about the setting? Is it always the same cost to try any target drug pairing, or are there different costs?
There can be different costs. For what we're going to talk about today, they're all going to be fixed costs, but that's because it's even bad enough to deal with that setting, right? But absolutely, we are going to have to deal with the cost-sensitive active learning, and also the oracle accuracy problem. So what we were talking about just a minute ago, although again, without knowing what the answers to those are. Yes.
To follow up on that, for this kind of experiments, is it true that you do one cell at a time when you do active learning, or do you, get multiple cells in a row?
Yep. So you can do either. One of the things that's kind of an interesting, what the response that biologists in general have had to this problem in large is to say, oh, we can't do one experiment at a time and get anywhere. Let's do 1,000 experiments at a time. Okay, like that's going to help. When you're talking about quadrillions or whatever, let's just do 1,000 at a time. We'll get there faster, but we'll all be dead anyway, right? And so the answer is that we have equipment that can do efficiently, more economically, sets of experiments, and so we do want batch-wise, in general, batch-wise active learning. Okay. Now, so as I was saying, most of the work that's been done has been done with retrospective studies. There are lots of problems with that. One of the problems is that we could be cheating without even thinking we're cheating by running the same analysis many times until we get the thing we're looking for, and also checking that it has meets the assumptions that we've made. But also, most of the work that's been done has been done with small such sets, because that's all that has typically been available. So here's some examples of databases, of data sets that have been used, and you can see that the number, this is the number of drugs and the number of targets, they're small compared to the size of the space we're interested in. By the way, there's also an interesting phenomenon here, which is, and we'll get back to this, that the question of how many of the combinations actually show a response, right? Whether responses are frequent or not. And many of these, the responses are quite frequent. But its data sets have been studied so far. Now, so here's the first result. We took a look at these data sets, and we did pretty standard uncertainty-based active learning, starting from nothing and building a model that I'll talk to you about in a minute, and showed, actually, I'm sorry, this was done with a basic matrix factorization method, but then with uncertainty sampling under that. And this is doing random learning, random choice, and this is doing active learning. And the point is that all of these data sets, where they had previously been only analyzed in a framework of give me 80% of the data and I'll try to then predict the remaining 20%, we were able to show that these were very good candidates for active learning, right? So you could do well in terms of building models quickly on this data. And that's just summarized here. If we take previous results on taking 80% and learning a model for 20%, and instead we do active learning up to 80%, we get much better accuracies, okay? But now we're going to move, yes?
Sorry, I had a question about that. You said the response rate is that like the number of non-zeros in the matrix?
Yes, exactly, yeah.
And you're saying it's...
Typically, that number is small. They are typically sparse. Yeah.
Oh, they are sparse?
Yeah, typically. But those data sets are not. Okay, in the real world, they're sparse, but in those, they're not. Right. Okay, so then now we were trying to then think about trying to extend this to something closer to the real problem. And of course, part of the problem is what data do we have? It turns out there's a database called PubChem, And there we created a subset of that data which had many more targets and many more compounds, okay? However, not all the experiments had been done. There was a lot of missing data from it. But there were features, chemical similarity features to use to measure similarities between drugs and targets, between drugs and between targets. Okay, so what we did then is compared, so here's the problem. Almost all of current matrix factorization type approaches don't work anything this big, right? At least not on computers we have access to. And so basically you can't do most of the things that have been done before on data sets this big, okay? Or you can't do it practically. And so what we looked at then was for tackling that problem, a problem where we did not have to construct the entire matrix and do operations on the entire matrix, but instead just did something very simple, which is to do, given the features for each of the drugs, make a lasso model to predict the target, and vice versa. Given all the features of the targets, predict responses to each individual drug. And so we have a whole bunch of row predictors and a whole bunch of column predictors, and we just average the results at each point. Okay? Very, very simple. But that's just as the driver for the active learning. That's the whole point, because So what we then did is compared random choice of experiments, and this is number of hits that were found in these sparse hits, or percentage of the discoveries. And that's the random, and this is the way that drug developers would have done this type of problem in the past, which is to build what's called a QSAR model, quantitative structure-activity relationship, which is a model for each drug, for each target alone, okay? And then so this is doing active learning with just that kind of model for just the targets. This is doing it with the combined model, and what you can see here is that we're starting to get the kind of results we want, which is we got 57% of the hits in this sparse matrix. It's 0.001% of hits, with 2.5% of the matrix experiments being done. So that's starting to be encouraging in terms of whether or not this is going to pan out. Now, of course, as I mentioned, this was based on having features to be able to make predictions even before we started. That turns out to often not be true. What do we do if we don't have features or they're not reliable? And also, that was all done with either binary or real valued outputs. What do we do if the outputs are multidimensional? So an example of that is much closer to the reality in the experimental work, is that we do an experiment by taking some cells, adding a drug, taking an image of the cells, and then try to figure out what happened, right? And so what it means is that we're now in the space where any phenotype is possible, right? And we don't know in advance what the phenotypes are. So we don't have anything. So what we did in this scenario was to say, okay, we're going to do some very straightforward things. We're going to measure features not of the drugs and targets, but of the image, and then make a representation of the possible phenotypes shown. So we're going to have image features like texture features, whatever you want. And then we'll have clusters of phenotypes that are going to be responses that are similar to different drugs that show the same response. Yes?
Very silly question, but so the images of the drug itself?
No, the images are of cells that are expressing a target treated with a drug.
Okay.
So that what's going to happen is the target is going to change when you add the drug. Okay? Yeah.
You mentioned, is there an implicit clustering assumption, or could things be just, on a linear assumption?
We're going to make an implicit clustering assumption, okay? But it's going to be done, as it turns out, we're going to do that on the fly, right? So we're going to keep clustering because we may have, in the last round of experiments, we may have learned a new phenotype, so we're continuously doing that, okay? So the point is, we're going to cluster the image phenotypes.
So it doesn't acquiring the images actually mean doing the experiments?
Yes, that is exactly. We don't have any images until we do an experiment. An experiment consists of exposing a target to a particular drug. Okay? Yes?
And just one other question about features. Does that include or encompass things like numerical simulations of whether this drug interacts or combined with this protein because we know what those chemical structures are? Is that part of?
It can be. Those simulations are expensive and not particularly revealing, or they're very good for cases where you have a lot of knowledge about that particular protein, many different structures of it and so on. So there are many cases where that's not very useful.
But in here, then, the features are basically just giving me some measure of similarity or relationship among different drugs and different targets.
Yes, and that was the previous setting we were talking about, where we did have such features. We're now in the setting where we don't have that. All we have is the images. We're going to make our own.
Gotcha.
We're using the same word. We're going to use our own features now of the images in order to decide what phenotype is being displayed. Okay, so now we're in a space where the thing we would normally think of doing then is to do some kind of matrix factorization on the phenotypes. And so we're going to do a seat of the pants type approach to doing that, which I'm going to tell you about, which is to say, okay, each experiment, we're going to assign a phenotype by clustering, which is a color. And then we're going to try to group the drugs by which drugs are not provably dissimilar. Okay? So we're going to group all the drugs that are potentially the same. So we're going to take the smallest set of groups that are consistent with what's known. Okay? And then we do the same thing for, well, I did that for targets, the same thing for drugs. Okay? And then the idea, of course, is that we can predict the phenotypes for everybody now, based on just assuming that if you're a member of this group of drugs, you're going to have the same phenotypes that any other member of that group has displayed. Okay? And that gives us a predictive model. Okay? So for anything where we at least have some measurements, we can do a predictive model of the responses. Okay? So how do we use that in active learning? All right, so this is also touching on themes we've heard the last 2 days. What we're going to do is we're going to say which experiments explicitly test the assignment of a drug to a group. Okay, so in other words, we're going to try to break the this implicit factorization we've done by grouping the drugs. Okay, and we're also going to take into account, if we were to falsify one of those assignments, how many other things would we have learned, how many other things would have been falsified? How many overall predictions would have been falsified? So this is, along the theme that's frequent in active learning of trying to get some sort of mutual information measurement, or some way of trying to measure the impact of doing an experiment on future models, okay? But without having the ability to do an explicit calculation of all of that, okay? All right, so then we pick a batch of experiments, we do the experiments, okay? Now, using that approach, we're going to now try to do this prospectively, okay? So we set up an automated robot that can add drugs to cells, and we set up an automated microscope that can take pictures of cells, and we set up computer pipelines to calculate all the image features and cluster the resulting features, assign phenotypes, build the model, pick the set. We're picking a hitting set of the experiments that we think maximize, the criteria that we just talked about. And then we're going to run that, and we just run it, and we're not going to get in the way. All right, so we're going to actually have the computer decide which experiments to do next. The robots do the experiments, give the data back, the computer decides on the next action, we just do that, okay? So here's the example of the space. So these are a bunch of different tagged proteins. So what I mean by tag is that we can see a specific protein in green when we take a picture on the microscope, and that tagged protein is changing. So these are 48 different tagged proteins that are going to be our universe of proteins. And we're also going to have 48 drugs, and we're going to try to learn the effect of those 48 drugs on those 48 targets, where we know nothing about either the drugs or the targets on their effects of each other. We know nothing starting out, right? So there's our space. But the problem is, since we don't know anything about this, we don't have any way of knowing whether anything's right. Yes?
So just maybe you touched on this, but I'm trying to figure out, so where are you getting, where are you either getting or growing the image features that you're clustering?
So the image, that's the thing that my group has been doing for a long time, which is automating analysis of this type of images. So we have a lot of experience with image features to analyze the phenotypes that are being displayed by the cells. Okay, so there are things like texture measures, distances between things, and so on.
Okay.
So we have a lot. We have a presume that you have a good. It's not actually a classifier because we don't have classes, but it's an unsupervised feature set. Yep. Okay. We have that. All right. So we don't know what the right answer is. So we can do all this work. We get some model at the end, but we don't have any idea of whether that model's right. So what we're going to do is we're going to duplicate all the drugs and all the targets, but not tell the computer that. So we're going to tell the computer there are 96 drugs and 96 targets, but there actually are only 48. And when it asks for 1 and 27, we're going to give it the same drug or the same target. So we know which are the duplicates, but it doesn't. Okay? So it's dealing with a 96 by 96 space that actually is 48 by 48. Whoops. Okay. Oh, okay. So we start out with all of the drugs. We just do the first round. We give it to start all the drugs for all of the drugs for. We start out with the no drug case for all of the targets, where we don't add anything. That's drug 1, or actually drug whatever it is, 36. Right? So we start out by that, and then it starts building its model. Its initial model, by the way, is that nobody has an effect. Right? But I know the phenotype of all the targets, but I don't know what, and everybody has no effect. And it starts choosing experiments. And now, when we stop, and we stop, interestingly, because we ran out of money. All right? We only had a certain budget for this. We ran out of money, we stopped. When we had done that, we had sampled 28% of the possible experiments. And that represented 72% of the drug target combinations when you take into account the duplication. Right? So as long as we'd sample 1 from every quad, we dealt with that one. So then we did the remaining unique combinations after the fact. So we did a last round where we just filled out the 48 by 48 space. Okay? And now here's the key thing. What we're going to do is we're going to ask at each round of active learning, how good was the model? But we can't compare the predictions because they're clustering dependent. So what we're going to do is we're going to ask how close was the predicted phenotype to the actual phenotype, okay? And in the feature space. And that's going to be our measure of the accuracy, okay? So here is, let's get this running. Here's a movie in which did it start? Oh, it started, okay. Purple means I'm wrong. Green means I'm right, the model's right. And white means we've actually done that experiment. And what you can see is, as you'd expect, as we're going along in the number of experiments on the bottom, we get more white, and the thing starts turning green. Okay? Yes?
Can you compare this, no, maybe not in this side of the matrix, but can you compare it to the best you could have done, so you compare it with the active learning tree? Like, what is the minimal subset you could need to get generalization to the entire matrix?
Okay, so one answer to that question is do all 48 by 48, right?
Well, all sub-combinations of them, right? Like it's a combinatorial, like for all of those, which subsets could you have picked so that they would have generalized?
Oh, right. If knowing at the end what the phenotypes are, what could we have clustered and what could we have gotten to, you mean?
Well, which subset was the minimal subset for the perfect active learning algorithm? So you've got some active learning algorithm, which is presumably somewhat myopic and not perfect. If an oracle could have said, these are the 10 ones to do, and that will actually generalize to your entire matrix. How many?
Right, not how many experiments it would take to get to that, but what would, if I just guessed the absolute right experiments, if I knew the right experiments, it's still pretty high. It's still pretty high because there's not a lot of similarity outside of the similarity we added by duplicating. Okay. Yeah.
Why am I seeing vertical purple strips?
Yes.
Why the arrows are that way?
Yes. So what that means is those are drugs that the model didn't learn well. And the reason is, it turns out, there are cases where the response of the cells is very variable. Okay? So in the number of experiments that we did, the number of images we collected per experiment, the phenotype itself was not robust. So that when we did the same experiment twice, we got two different apparent phenotypes, and therefore, of course, the model got confused. Right?
So this is unique to your application, right? It's not general.
Well, right. In this case, we did not add in, do more experiments until you get, a more robust error, right? We just had a fixed number of experiments. But that would certainly be that's Rev 2, right? Okay, so what is the overall picture here? After 28% of the experiments, the model's 92% accurate. Yes?
So just quick calculation. So 48 squared is like 2,500 or something. And then on the previous graph, you were showing many more experiments, and yet there were, the graph, the matrix wasn't white, right?
Right. So the space is 9,200, right? 96 by 96. Right, because we duplicated everything, but it doesn't know that.
Oh,
Okay?
oh, and you're just showing this, okay.
Yeah?
Okay.
All right, so 28% of the experiments, 92% correct model, and random choice of experiments would have gotten us about 40% less accuracy. Okay? And by the way, almost all of that performance came from it figuring out quickly the duplication structure, which of course is exactly what we're hoping in the real world, that it's got to find things that are similar, right? There's no free lunch. The only way it's going to be able to do that is to find things that are similar. Okay, so last thing I'm going to just talk about then. So this, by the way, is the first example of active learning in biology where we didn't know ahead of time what the phenotypes were. We didn't know anything. Okay, so it's the open-ended, real-world case that we're going into some space and we're just going to start doing experiments to try to get answers. But in that real-world theme, one of the real issues is when do I know to stop, right? Assuming I still have money. Should I stop and save some of that money? And this is, there's work on this problem. It's surprisingly not talked about very often. And that's probably because so much active learning work is done retrospectively where you know the final full model, and so you can calculate the accuracy and you can do everything, right? So, but in the real world, of course, the whole point is to avoid doing those all the experiments, and you don't have an estimate of the overall accuracy other than what you get from what data you have, all right? So we need a stopping criteria, stopping rule or something. Typically in the past, there's been proposals for a single criterion like, how consistent are the predictions, or something like that, okay? And we just decided to try a different approach to that I'm going to talk to you about. And in order to do that, what we're going to talk about is the idea that we can characterize an experimental space, all right? And we're going to characterize or parameterize an experimental space using, in our case, two parameters, which are the sparseness of the interactions, that is, how often does a target respond to a drug, right? An arbitrary target respond to an arbitrary drug, what's the probability that it will affect it, and how similar the drugs or the targets are to each other, right? So if you get a uniqueness of 100%, that means there are no factors, it's full rank, in which case go home, right? But those are going to be that's our theoretical description of the experimental space. I'm not going to defend that one over others, but the idea is I'm going to create some parameterization of the experimental space. By the way, there's another hidden parameter in here, which is the phenotypes, how many phenotypes there are, all right? But we're not going to worry about that right now. So what we did is a whole bunch of experiments using the algorithm I just talked about before, this no features, empirical clustering and factoring. And this shows the improvement of active learning over random depending on the responsiveness and the uniqueness. Right? And as you expect, if you're in the unique case, it's hopeless, right? You're getting no improvement. And if you're in the case where there are very, very few responses, you're the needle in the haystack case, and you're not going to do too much better there. Oh, you might be lucky. But in the big swath of this, we're getting significant improvement, of course, with active learning because there is something to learn. There's some factorization to learn. There's some grouping to learn. And you have enough phenotypes occurring that you find them because you have to find some phenotypes in order to be able to group the compounds, the drugs. Okay, so assuming we have some such parameterization, what we're going to do is we're going to define a set of features of an active learning trajectory. Okay? So if we do a simulated, by the way, all of these were simulated results. Okay? This is, we presuppose we have a model of this uniqueness, this responsiveness, synthesize data for that, then do active learning on that data, and see how well we did compared to random. Yes?
Just the bottom row there is because it's so low dimensional that you can just take random samples and learn it?
Yep. And so this we can do. We can do the simulations, whatever the parameterization of the space is, we can subdivide it and do simulations for the whole space, or a big portion of it. And what we're going to do then is for each of those active learning curves, each of those learning curves, we're going to calculate features of the curve. Like, how did the consistency change from step to step? How many things did I find so far, and so on and so forth. All right, there's a whole bunch of them. But then what we're going to do is learn a regressor from those features to the accuracy, because we know the correct accuracy because it's simulated data. So what we're going to then do is to say, if this is a fair parameterization of the space, then I will have a predictor of accuracy for experiments drawn from that space. Okay? And so that's what these results are. This is showing the correct accuracy versus the predicted accuracy. And this is the probability that the real accuracy is higher than my predicted accuracy. So this is, I'm estimating a lower limit for the accuracy. And for this whole space here, I can do that, right? It's only when I get all the way up to like 95%, saying that the accuracy is actually higher than 95%, of course, it's the probability of that's dropping off, okay? So in the range that we probably care about, 80, 90% accuracy, I can get, at least for these experiments, very accurate prediction. Does that make sense? Okay. And so then we did that for retrospective data that we talked about earlier, all right? So this is the actual learning curve, and this is our estimator, which is being conservative, saying, all right, I think this is the correct accuracy, all right? And so it works for all, for each of these cases that we get. And if we do the same thing we did before, now I've changed this column. This is not doing 80% training, 20% testing. This is deciding to stop after 50 or 30 or whatever % of the experiments because I think I'm done, all right? And I can get a comparable accuracy to having just done the 80% by stopping automatically with that rule, okay? So the idea there is just that because in the real world cases where we know so little about where we're working in, these are potential tricks or tools that we can use to try to adapt ourselves to that space by using this idea of simulating experimental spaces. Okay? So, yeah.
So in this simulated world, when you kind of try to design this prediction by looking at the learning curve or whatever, are you implicitly sort of taking a uniform average over that space? And is that...
So what's happening is each simulated active learning experiment gets a set of features, and they get thrown into the regressor, and it just tries to estimate an accuracy for that. Right? Well, they get associated with an actual accuracy that you ended up with.
So
right.
Every one of those points in that 2x2 set is sort of equally weighted.
It's being equally weighted thrown in. We could obviously do better than that, but that's the start. Okay. Now, and one of the big factors there are being able to have some such parameterization, and then we run it on the data for the data that is matched to whatever space we're actually going to try to work in. Okay, so conclusions: we have some results on active learning, both from retrospective data sets and for this prospective data set, where we're starting from very little, which is the real range that we're interested in biology. Done this in terms of the first real use of this for unknown phenotypes, and I've talked to you about this idea for trying to estimate accuracy empirically. Okay, so these are the people that did most of the work. And people that funded it, and I'll stop there, and thank you very much. We use up all the questions? Yeah.
So it seems something odd to use raw accuracy as a measure. I mean, presumably, like, predicting, unexpected adverse interactions is much more important than predicting the vast majority of zeros in the matrix. And so you've looked at compensating for that? So
it's a great question. For interest of time, I flipped around between accuracy and area under the curve, which takes that into account, right? So in other words, in some of those, I was measuring AUC as...
Sure. But even with AUC, right? I mean, in some interactions, maybe they're obvious, and so you wouldn't, you know they exist anyway, and some are unexpected, and therefore much more important to catch.
So we can have a long conversation about my philosophy to computational biology, which is most of what I learned when I was a PhD student at Caltech in biology 40 years ago is wrong. Okay, so expected, not expected, not really important concepts to me. Okay, in that. I just found out about one that was discovered a couple of years ago that I didn't even know about, which is that you can make proteins without ribosomes, right? I mean, the whole idea was supposed to be, DNA makes RNA, makes protein, and protein is made by ribosomes from RNA. Well, you can just skip the ribosomes, and you can do it by this totally other system that nobody knew about for, decades, right? So I think that's the answer to your question. But I certainly could put in, if I wanted to, well, I already know these are, major interactors, and so I'm going to, I have to put more weight on that or something. I could try to do that, but yeah. Okay, great. All right, time for a break.